Все вімкнено, ви можете розпочинати.
Доброго дня, шановні колеги. Сьогодні 11 липня 2024 року, 12 година. Отже, dear colleagues, today is the 11th of July 2022, 12 o'clock Ukrainian time. Дозвольте розпочати засідання. Відповідно до рішення Вченої ради Сумського національного аграрного університету від 18 грудня 2023 року протокол номер 7 та відповідного наказу ректора по Сумському національному аграрному університету утворено спеціалізовану вчену раду для прийняття до розгляду та проведення разового захисту дисертації ЧЄНЬ ЦЯОЯНЬ селекційно-генетичні основи ознак колосу пшениці озимої на здобуття ступеня доктора філософії в галузі знань 20, аграрні науки та продовольства за спеціальністю 201 агрономія. According to the discussion of the Academical Council of the Senior National Agrarian University, Protocol number 7 in Rector Orders in our university to accept to consideration and conduction on time online defense of ЧЄНь ЦЯОЯНЬ dissertation, green and genetic basis of winter wheat earth race. The knowledge field 20, agricultural sense, and food specialty 201, агрономія. Згідно реєстраційної карти, у нас має бути присутні 5 членів спеціалізованої вченої ради. Голова ради Мельник Андрій Васильович, доктор господарських наук, професор кафедри сільської партії лісового господарства Сумського національного аграрного університету. Тобто я присутній. Рецензенти: Бутенко Андрій Олександрович, кандидат сільськогосподарських наук, доцент кафедри агротехнологій та ґрунтознавства Сумський національний аграрний університет. Присутній?
Так, присутній.
Так, є, ми фіксуємо. Верещагін Ігор Володимирович, кандидат сільськогосподарських наук, доцент кафедри селекції та насінництва імені професора Миколи Гончарова, Сумський національний університет. Присутній? Так, підписуйтеся. Офіційні опоненти: Маренич Микола Миколайович, доктор сільськогосподарських наук, професор кафедри селекції та насінництва і генетики Полтавського державного аграрного університету. Микола Миколайович, засобами Zoom-конференції.
Присутній.
Дякую. Фіксуємо. Далі. Леонов Олег Юрійович, доктор сільськогосподарських наук, старший науковий співробітник, завідувач лабораторії селекції пшениці Інституту рослинництва імені Юрєва Національної академії аграрних наук, теж засобами Zoom-конференції присутній. Олег Юрійович, включіть мікрофон і скажіть.
Я єсть.
Так, дякуємо. Тобто у нас є кворум. Ці члени спеціалізованої вченої ради присутні сьогодні на засіданні. Прошу зареєструватися, але ми вже з Ігорем Володимировичем зареєструвалися, а інші засобами відеоконференції я вас зареєстрував. Отже, є всі підстави розпочати роботу спеціалізованої вченої ради. Таким чином, засідання спеціалізованої вченої ради оголошується відкритим. На порядку денному сьогодні захист дисертації аспірантки денної форми навчання селекції та кафедри селекції та насінництва імені професора Миколи Гончарова ЦЯНЬ ЦЯОЯНЬ на здобуття ступеня доктора філософії 201 агрономія. Тема дисертаційної роботи селекційно-генетичні основи ознак колосу пшениці озимої. І коротко про етапи виконання науково-навчального навантаження до освітньої наукової програми. Так правильно. Роботу виконано в Сумському національному аграрному університеті та Фінансовому інституті науки і технології. Китай. Науковий керівник Кандиба Наталія Миколаївна, кандидат біологічних сільськогосподарських наук, доцент кафедри селекції насінництва імені Миколи Гончарова, Сумський національний аграрний університет. До спеціалізованої вченої ради надійшли такі документи. Ось вони в мене всі на столі. Відповідно, копія першої сторінки паспорта громадянинки Китайської Народної Республіки, копія диплома магістра, витяг з наказу про зарахування, академічна довідка про виконання відповідної освітньої програми, висновок наукового керівника доцента Наталії Кандиди, висновок про наукову і практичну цінність, підписаний нашим проректором наукової та міжнародної діяльності Юрієм Демком, дисертація в електронному та друковому вигляді, відбитки наукових публікацій, які пропонуються за темою дисертації. So, we have all documents that we need for our meeting. All documents here, some documents with stamp, with signature, so everything clear. We can start next step. Всі документи відповідають вимогам. Дисертація до захисту подається вперше. Таким чином, хто за запропонований порядок денний, прошу голосувати за проведення роботи спеціалізованої вченої ради до захисту дисертації, яка була проведена перед цим. Хто за? Так, ми скрізь не робимо, в нас це є свідомо. Чудово. Таким чином, всі за, хто проти, немає, утримався, немає. Одноголосно. Таким чином, дозвольте зазначити біографічні дані аспірантів. So, I will tell some information about biographic information the PhD student. ЧЄНЬ ЦЯОЯНЬ, 1989 року народження, громадянинка Китайської Народної Республіки, освіта вища. У 2018 році закінчила Фінанський інститут науки та технологій Китайська Народна Республіка та здобула освітній ступінь магістра в спеціальності генетика і селекції. На теперішній час працює в коледжі програмного забезпечення Інституту науки і технологій Фінань на посаді секретаря педагога. Аспірантуру закінчила в 2022 році в Сумському національному аграрному університеті Міністерства освіти і науки України. Місто Сум. Роботу виконано в Сумському національному аграрному університеті та в Фінанському інституті науки і технологій. Тема була затверджена на засіданні вченої ради Сумського національного аграрного університету, протокол номер 7 від 26 грудня 2018 року та перезатверджена на тему селекційно-генетичної основи ознак колосу пшениці на засіданні вченої ради протокол номер 12 27 лютого 2023 року. У 2022 році ЧЄНЬ ЦЯОЯНЬ завершила виконання освіти наукової програми. 2 лютого 2023 року відбулася публічна презентація дисертаційного дослідження на кафедрі селекції насінництва імені Миколи Гончарова за спеціальністю 201 агрономія, де дисертацію було рекомендовано до розгляду та захисту у разовій спеціалізованій вченій раді. Основні положення отримані наукового результату роботи висвітлено у 12 наукових роботах, яких 2 видання, що індексуються в науково-метричних базах Scopus та Web of Science, 3 у наукових фахово-академічних виданнях, 7 наукових міжнародних конференцій. Згідно вимог пункту 8, 9 порядку присудження доктора філософії, дані публікації відповідають вимогам, що підтверджуються рішеннями безпосередньо порядків та наказів. Далі у нас іде інформація щодо самої, чи є питання спеціалізованої вченої ради по інформації про здобувача. Так, немає питань до здобувача. Далі, згідно протоколу, ми маємо заслухати доклад нашого здобувача. So now, ЧЄНЬ ЦЯОЯНЬ. If you're ready, you can start after 1 minute or maybe 10 seconds. Світлана Васильівна зараз, мабуть, включить нам доступ до презентації, і вона потім почне працювати, доповідати свою презентацію. Світлана Васильівна. ЧЄНь ЦЯОЯНь, can you start your presentation?
Okay. Thank you.
Just 1 moment, please show your screen.
Okay. Dear Chairman, dear members,
No,
our
stop,
special
stop,
guest.
stop, stop. We cannot see your presentation. Just 1 moment. Just 1 moment. We cannot see your screen. Шановні колеги, ви теж не бачите екрану, бо ми бачимо тільки нас усіх. Це аспірантура виключила презентацію, тепер вона тільки може включити її. Тобто вони зробили нам послугу, але не проконтролювали до кінця. Зараз чекаємо.
Beginning.
Yes, you can start.
Okay.
Please start your presentation. But
Okay.
please start, make presentation in your screen.
Dear Chairman, dear members of Specialized Council, dear audience.
No, stop. We not see your screen. Скажіть аспірантурі, хай поверне все, як було.
What?
No, don't worry, it's not.
Андрій Васильович, доступ до поширення екрану наданий. Він був наданий з самого початку.
Зараз, тоді ви виключили її презентації, зараз вона
Так, для того,
не
щоб
може.
всіх членів ради було видно, і зараз
Так.
вона має знову поширити свій екран.
Ну, вона робить і каже, що не виходить. Окей, Jen, can you start again? So, start again. Start your
Okay.
presentation. Not your speech. Presentation in PowerPoint.
Okay.
Okay. Please restart. Make presentation in PowerPoint in your screen. Can you see your presentation in screen? І поки вона перезагружає, так поняв, що вона зрозуміла, що робити в такій ситуації. Шановні колеги, взагалі нас нормально чутно? Микола Миколаївич,
Чути чудово.
це. Бо я вже, бачите, біля мікрофона вже на дисертації його поставив, вже все. Jen, what about your presentation? Can you start in your screen? Make full screen presentation and show for everybody.
Okay.
Okay, please start in your computer. Can you see
Okay,
now?
okay,
in
okay.
this-
Okay, I see.
Щось получилось. Yes, okay, we can see just your screen, not presentation. Okay, now it's very clear. Okay, very good. We see first page, so you can start now your speech and change the slide, please.
Okay. Start.
Yes,
Dear Chairman, dear members of the Specialized Council, dear audience, the relevance of research in this field is underscored by several key factors. But one, by 2050, the global population is projected to reach 9 billion. This will significantly increase the demand for food, necessitating a more than 30% increase in the production of cereal crops, including winter wheat. 2. Traditional methods of weeding are time-consuming and resource-intensive. 3. China is the world leader in terms of both wheat acreage and total production. Therefore, the increasing yields in China can have a substantial impact on global food security. QTL mapping of quantitative to the local based on genetic mapping of molecular marker is an effective technical tool for improving wheat yield. 5. Yield is a quantitative rate that is controlled by managers and depends on various environmental factors. And therefore, the use of the modern methods of genetic analysis allows for more accurate identification of genes that affect yield. The purpose of the study is to identify the molecular genetic basis of winter wheat productivity traits, in particular, the structure of the ear, as well as to develop and implement marker-assisted breeding methods to increase yields and improve other agronomic characteristics of winter wheat. The object of the research is the genetic composition of 3 yield factors of winter wheat and the regulation of mean MRS in development. The research subject is genetic local regulating the number of spikelets, mechanisms of growth, and the development regulation by MRS. The study was conducted at the environmental field and the laboratory of the Henan Institute of Science and Technology. These parent forms have unique characteristics: high yields, resistance to major disease of the shepherd's pulse, high protein and gluten content, good adaptation to different climatic conditions and soil types, and resistant to drought and high temperatures. This variety is Mexican largest spike, more gaze, but low weight of 1,000 gaze. This variety is Bainon 4199, is high 1,000 gaze weight, but low gaze per year. In this study, we established the F2 population of Mexican largest spike and Bainon 4199 and conducted a preliminary QTL mapping for spike street, such as gain number per year and 1,000 gain weight, using SSR molecular markers technology to lay the foundation for accurate mapping of target QTL, cloning of the relevant genes, and development of the molecular markers in the future. The results of the research showed that the sequence of with SR primers was evenly distributed among the 3 wheat chromosomal cluster A, B, and D. In total of the 2 pairs primers were collected and synthesized by Shanghai Biotechnology Synthesis. Regarding the analysis of phenotypic data for spike traits, the population was phenotypically highly variable for both gain number per spike and 1,000 gain weight. The results of the population normally analysis showed that these 2 traits normally distribute with bidirectional superparent segregation, indicating they were inherited as quantitative traits. Correlation analysis as that gain number per year and 1,000 gain weight were highly significant and positively correlated with a correlation coefficient of 0.953. SSR marker polymorphism screen and polymorphism amplification in the F2 population revealed that a total of 143 distinctly banded polymorphic marker pairs were identified, which was screened for molecular marker polymorphisms using 300 SSR primer pairs for the F1 between the Mexican larger spike and the binom 4199. 143 pairs of polymorphic markers were genotyped and genetic maps were conducted for 147 individual plants of the F2 population, amplified bounds of some polymorphic marks in the parent population. A negative QT analysis of gain number per year and 1,000 gain weight. These results showed that a total of 10 educational QTs associated with gain number per year, and 1,000 gain weight and spike number per year were identified, which could explain 4.92 to 21.10% of the phenotypic variation. The 10 additional QTLs are mainly distributed on the following columns. QTL analysis showed that 6 epistatic QTL local associated with gain number per year mainly on chromosomes 2B, 2D, 3B, and 6B. And the effect values were smaller for 0, which confirms the genetic effect of epistatic QTL local, which belongs to the recombinant type, and which could explain 41.91% of the total phenotypic variation. 3 epistatic QTL local associated with 1,000 gain weight, mainly on chromosome 3B and 5A, among which the genetic values of these two genes were greater for 0. Belonging to the parent type, this indicates that there are some random local on the chromosomes that don't directly affect the phenotype, but this local can affect the phenotypic traits by interacting with each other. Phenotypic analysis of the developing is again an important period because it determines gain yield and quality decrease for weight harvesting. Based on the results of research, weight gains at 7 and 14 days after pollination were isolated for the identification of miRNAs involved in the development of weight gains. Weight gain development was assessed by observing patterns of gain weight and volume gain, and to determine the relationship between these changes. We used the small RNA libraries and datagrams from 7 and 14 days after pollination gains to compare mean RNA expression. The results of the sRNAs sequencing studies in early gain development revealed unique and common sRNA sequences between 7 and 14 days after pollination. As can be seen from the table 4, the 7 and 14 days after pollination contained 33.69 of the total sRNA number, but only 8.3% of the unique sRNAs, and the total number of the unique sRNAs in the 7 days after pollination library is less than the 14 days after pollination library. The results that compared with 7 and 14 days after pollination get a broader sRNA-mediated regulation of gene expression. Regarding the results of annexation and the distribution of sRNAs in libraries from 7 and 14 days after pollination gains, after removing unwanted RNAs, the number of remaining unannounced sequence was 17.46 in 7 days after termination and 33.3% in the 14 days after termination libraries. The results of SR length distribution in developing at 7 and 14 days after termination established that MR length distribution generally reflects species or tissue specificity per RNA ranged in length from 8 to 30 NT in both libraries, with a majority between 21 to 24 NT in length. The most common length is 21 in 7 days after termination, and the 24 NT is in 14 days after termination library. Regarding the summary of the length distribution of clean race and microRNA by sequencing, different lengths from length distribution of clean race and mRNAs were determined in the 2 libraries. The length distribution mRNA was 61 and 73.89% in 20 to 24 NT, respectively, and the percentage of the mRNA with a sequence in the of 24 NT in 7 and 14 days after termination was higher than other sequences. To predict with novel mRNAs, unannounced mRNAs were analyzed based on pre-mRNA sequences that can form a canonical hairpin stem loop structure. A total of 79 novel mRNAs were predicted, of which 32 were expressed in the 7 days after termination library, and 38 were expressed in the 14 days after termination library. Identification of known mRNAs and prediction of new mRNAs. Regarding the expression of known mRNAs, including 4 genes, they were expressed most actively. And this 6, but other genes 6 in moderate abundance. Most of the abundantly expressed mRNA were known mRNAs from the wheat mRNA database and conserved between plant species, such as this genes and this genes. Further results of verification of partially differentially expressed microRNAs using qRT-PCR, it was found that the expression patterns of these microRNAs correspond to those obtained from deep sequencing, indicating the reliability of mRNA sequencing. Plant mRNA generally show performance and their target genes with important functions, predict the target sequence of all microRNAs, and difference expression microRNA was searched mass in COG, GO, KGG, Swiss Prot, and NR database. 810 targets from the total number of microRNAs and 477 difference expression mRNAs targets were able successfully obtain announced functional information. Of all 810 microRNA targets, 174 and 605, 176 can be respectively announced in the COG, GO, and KGG database. Based on the results of gene-specific qRT-PCR testing, 8 target genes of microRNAs, 5 known and 3 novel. And the result shows that at 7 and 14 days after plantation, the expression of these two genes and their target genes is negatively correlated. These two genes and its target genes is positively correlated, and this genes is no clear correlation. There is likely to be complex temporal and spatial regulation between mRNAs and their target. Dear President, since the conclusions were announced during my report, let me not read them out, but go straight to the proposals. Hybrids obtained from crossing the parental forms, Mexican Lady Spike and Bino 4199 were recombinant and transferred. Using the breeding practice and for breeding testing of winter wheat in Henan province, Henan wheat consortium for the validation of wheat varieties for yield, disease resistance, resistance to lodging, and quality, we will contribute to the further selection of high-yielding varieties of winter wheat. Those conclusions are from my report. Thanks everyone for attention. My report is finished. Thank you.
Thank you. You're exactly in time. We spent exactly 18 minutes. Okay, dear colleagues, the report is finished. Let's go on our meeting. Dear members of council, are there any questions? Please ask some. So, dear colleagues, please ask some questions. Шановні колеги, задаємо запитання згідно процедури. Маємо задати по презентації запитання. Можливо, Микола Миколаївич, він там неможливо, він же руку підняв, але я б ще звук включив. Окей, професор
Звичайно,
Микола
я ж
Маревич.
спочатку, як дисциплінований член разової спецради, включаю руку, а тоді включаю голос.
Thanks a lot. You really good in our document. Okay, please.
Thank you, Andrei Vasilievich. Chen, are there any advantages of marker-assisted selection over classical breeding methods? And the second question, what principle underlies the constructions of the genetic linkage map in your research? Thank you.
So,
Okay.
Jen, yes, please give answers for these two questions.
Dear Mikhail Lvovich, thank you for your questions. Let me provide the answers. Question 1. Are there any advantages of marker-assisted selection over classical breeding methods? My answer. Marker-assisted selection has several advantages over classical breeding methods. Firstly, it allows for faster and more accurate identification and selection of plants with desirable genetic traits, reducing the time needed to develop new varieties, and minimizing the risk of errors in the breeding process. Marker-assisted selection is also effective for the selection of complex traits controlled by multiple genes, which is difficult to achieve with classical methods. Additionally, it helps maintain genetic diversity, and in the long term, marker-assisted selection can be economically beneficial by reducing the time and resource required for the breeding process. Question 2. What principle analyzes the construction of the genetic linkage map in your research? My answer. The principle underlying the construction of genetic maps in our research involves using molecular markers to determine the positions of genes and chromosomes. This process includes analyzing associations between markers and phenotypic traits, allowing us to establish genetic linkage and map gene locations on the plant genome My answer is finished.
Okay. Jen, next time, please not tell again the question. Just give answers. Mikola Mikolayevich, are you satisfied for these questions?
Yes, I am satisfied.
Okay, thank you very much. Okay, next, colleagues. Yuri Olegovich, do you have any questions? Doctor Agricultural Science, head of which breeding laboratory, Institute, Plant Production, National Academy of Agrarian Science of Ukraine. Oleg Yurievich, can you hear us?
Yes, thanks. I have some questions. What is the genealogical origin of Mexican large spike form, and where did it come from? Because I could not find the samples in CIMMYT database.
Dear Oleg Yulievich, thank you for your questions. Let me provide the answers. Question one of my answer. Mexican large spike was developed in Mexico and is known for its large spike size. It has a genealogical origin from the Mexican wheat variety Tonnery as a mother form and a father form that was obtained by selection from local wheat populations in Mexico. And the question two of my answer.
Юрику, я на засіданні, ніяк говорить. Ага.
Extrapolation of the detected influence of the presence of certain marks on the phenotypic manifestation of protective traits in other weather and ecological and geographical conditions, especially stressful ones, is a complex task that requires a multidimensional approach. However, we can conduct field trials in different regions and with different climatic and environmental conditions, use models that take into account climatic and environmental variables, identify genetic mechanisms of stress resistance and adaptivity, and assess interaction between genotypes and the environment, which will allow us to understand how certain genetic conditions manifest themselves in different conditions. My answer is finished. Thank you.
Okay, excellent. Very good answers. It was very good questions. Okay, next. Who will next? Maybe Andrei Vitenko.
Chen, I have two questions. The first question, what spike traits contribute to the yield of winter wheat? And second question, why were SSR markets specifically used?
Thank you, dear Andrei Alexandrovich. Thank you for your questions. Let me provide the answers. Question one of my answer of the question one. The spike traits that contribute to the yield of winter wheat include the number of spikelets per spike, the number of gates per spikelet, gate weight, spike length, spike density, spikelet fertility, and larging resistance. My answer of the question two. For the reasons, the use of SSR markers is justified due to their universality, high polymorphism, and ease of the analysis, making them essential tools for molecular genetic studies in breeding. These markers are characterized by their high polymorphism and genome-wide distribution, ease of determination through polymerase chain reaction, and geo-electrophoresis phenol analysis, genetic stability, and wide application in breeding to identify genetic markers associated with desirable phenotypes. My answer is finished. Thank you.
Microphone? Okay, thanks. Okay, good, very good. And other colleagues have some questions. Either in Shahin, please ask some questions. Okay. With the interpretation of such a field that will develop certainly all recommended by NATO Minister. It's first one, it's and please explain the mechanism of action of the molecular matter related to the target immunity. Okay, yes, okay, Jen, please give the answers for this question.
Dear Igor Dimilovich, thank you for your questions. Let me provide the answers. My answer of the question 1. Usually, the manufacturer's recommendations are used, but sometimes the amplification program is adjusted according to the concentration of the template. DNA and the size of the fragment PCR reaction conditions are mainly temperature, time, and the number of the cycles. We have optimized the annealing temperature and extension time recommended by the marker manufacturers ourselves, because the annealing temperature and time depend on the primer length. Based on composition and concentration, the length of the target base sequence. My answer of the question 2. The mechanism of action of the molecular marker in relation to a target J can be described as follows. First, a marker is selected and the marker can be a specific DNA sequence. Secondly, amplification of this DNA sequence. Third, analysis of the amplified DNA. Fourthly, the results of the marker detection are compared with the phenotypic characteristics. And if the marker is constantly associated with a particular phenotype or gene variation, this may indicate that the target gene is located in the vicinity of the marker. My answer is finished.
Okay, thanks. Okay. Do you have any more questions, dear colleagues? If somebody have more questions? No? Okay, I have also some questions. Dear Jen, I have some questions. First of, it's like, is it some new sources material obtained for the winter breeding? Is some new sources material obtained for the winter breeding? It's the first question. And the next one, if new material has been obtained, what is the future prospect for working with each? So it's two questions with similar direction.
Dear Andrei Vasilyovich, thank you for your questions. Let me provide the answers. My answer of the question 1. Several new lines were obtained by crossing two parents, Mexican large spike, the binom 4199, and back crossing and recombinant self-linked lines in the resulting progeny after 4 to 5 years of selection. My answer of the question 2. The new lines created have been submitted to the winter wheat breeding trial and to the Henan Consortium for wheat variety variation based on yield, distance, disease distance, loading distance, and quality. Once all these indicators are achieved, the new wheat variety will be widely promoted and planted in the region where it is adapted, increasing farmers' income, as evidenced by a certificate of conformity. My answer is finish.
Okay, thanks. I want to ask again if some colleagues have more questions, maybe more detailed questions. It's enough. It's enough. Yes, 10 questions, it's enough. Okay, now in our procedure, I must tell you some information from our university. It's like висновок про наукову та практичну цінність. Тобто я маю зачитати її на нашій державній рідній мові, тому що вона завірена печаткою, буде передавати Міністерство освіти. Отже, висновок про науково-практичну новизну, теоретичне значення, результатів дисертації, аспірантів кафедри, селекційних професора Миколаєм Миколаєм, ЧЄНЬ, названі аграрні науки, продовольства. Тобто це згідно, по якій процедурі ми зібралися. Актуальність. Пшениця озимна була і залишається однією з важливих зернових культур, що здатна забезпечити продовольство населення планети. Чисельність якого постійно зростає. Наведена актуальність, наведені методи сучасні, генетичні, селекційні, які використовуються в даній дисертаційній роботі. Також зазначений зв'язок роботи з науковими планами, зокрема план Київського національного аграрного університету, вдосконалити елементи зернових культурних культур з урахуванням оптимізації агротехнічних умов та агродосвідконтролю, розвитку, в тому числі Міністерства України, номер державної реєстрації. Наведена наукова новизна, що вперше було досліджено генетичний механізм кількості зерен, колосів пшениці з використанням екстремальних різниці в кількості зерен двох батьків. Це Mexican Invasion 419. Була створена карта генетичного щеплення пшениці, що містить 143 молекулярних маркерів, що охоплює 19 споробних впливів. Також наведено, що теоретичне практичне значення даної роботи, що вона буде використовуватися в навчальному процесі, має практичне значення для генетики, селекції і навчання. Особистий метод полягає в одержанні наукових та практичних результатів, що виконані в роботі, в аналізі літературних джерел, наведенні напряму, мети. Потім результати висвітлюються в наукових публікаціях, часто авторство, які складає від 20 до 100%. Як ми вже зазначали, є такі скопусі, і також категорії того, чи відповідає чинній вимогам. Характеристика здобувача, вона пройшла шлях від навчання в фінансовому університеті за спеціальністю генетика селекція, на навчання в аспірантурі Київського національного аграрного університету, виконала програму, просили довідки, тож сьогодні наводила документи присутні. Ще раз зазначаємо, що всього 12 публікацій, із них 2 статті в описі, 3 статті в видання. Ці наведені з посиланням, тому вони є наведені на нашому сайті, і якщо хтось захоче з ними знайомитися, то можна зробити. Ну і як висновок, вважати, що дисертаційна робота ЦЯНЬ на тему селекційно-генетичної ознаки основи ознак колоса пшениці за рівнем актуальності досліджень, обґрунтованості та практичних значень відповідних результатів, наукові новизни та високому рівні впровадження системи виробництва, посилання на відповідні пункти Кабінету міністрів, постанови наказу Міністерства освіти, відповідає. Рекомендувати дисертаційну роботу селекційно-генетичні основи ознак колоса пшениці озимої, виконані ЦЯНЬ на здобуття доктора ім'я, і є підписи рецензенти, доцент кафедри технології Андрій Бутенко, та доцент кафедри селекційного Ігор Верещанин, і завідувач кафедри, який був головуючим на даному засіданні, кандидат професорських наук, доцент, завідувач кафедри селекційного Віктор Аничко. Також даний документ підписаний проректором Юрієм Банком. Отже, виконана дана вимога є. Далі, згідно процедури, Переходимо до розгляду дисертаційної роботи. Запрошені виступи рецензентів. So, advanced in the procedure, let process to consider of the dissertation work by ЧЄНЬ ЦЯОЯНЬ. I invite the reviews to speak. First, it can be Андрій Бітенко. Андрій Олександрович, you are welcome.
Дякую, Андрій Васильович. Зачитувати всю рецензію чи тільки ті моменти, які окремо виділені?
Побажання,
Зауваження.
які ви хочете. Як ви вважаєте, можете трішки про методику, актуальність і відповідно, які ваші побажання. Окей?
Добре, ну, в будь-якому випадку рецензія, вона оприлюднена на сайті, просто щоб не розтягувати час. Дякую. Review of the official reviewer Андрій Олександрович Бутенко, PhD of Agricultural Science, Associate Professor of the Department of Agricultural Technology and Soil Science of Sumy National Agrarian University. For the PhD thesis, ЦЯОЯНЬ ЧЄНЬ, Breeding and Genetics, Genetic Basis of Winter Wheat Traits, submitted for Doctor of Philosophy, Scientific Degree, Field of Knowledge, 20 Agriculture Science and Food Specialty, 201 Agronomy. Relevance of the dissertation topic. Winter wheat is one of the world's three major grain crops, and China ranks first in the world in terms of both area planted and total wheat production. According to the predictions by 2050, the world population will increase from the current 7 billion to 9 billion, and the food production will have to be increased by more than 30% in order to meet the increasing consumption demand of human beings. Therefore, the cultivation of new varieties of the three major food crops, namely high yielding and stable yielding wheat, rice and maize, is an effective way to satisfy the future dependent on human beings for food. Yield is quantitative trait controlled by multiple genes with complex genetic basis and is usually influenced by the environmental. Utilizing traditional breeding methods is time and resource consuming. However, with the development of molecular biology and biostatics, QTL mapping of quantitative traits loose basis based on genetic mapping of molecular markers provide and effective technical means to study the genetics of quantitative traits. The next, the main results of obtained personally by the author. A map of the genetics linkage of wheat was created, which contained 144 molecular markers, spanning 19 wheat chromosomes with a total genome length of 3,128 cm. The average distance between the markers is 25 centimeters, and the minimum genetic distance of 40 centimeters. number of scientific publications. The main items of the thesis are presented in 12 scientific medical scopus and science, 7 abstracts of reports at international scientific and practical conference and one it is to provide
Андрій
fields
Олександрович, ми вас погано чуємо. Якщо можна, переходьте до висновку.
Ага, добре. Correspondence of the так, чуть-чуть, так? Of
Так, так.
the to the especially the word. Of ЦЯОЯНЬ, of interviewed submitted for degree of philosophy in the field of political science and food in the special according level main provision the results in professional public knowledge of the proposal is practical scientific the dissertation and the resolution of the
Окей, окей. Андрій Олександрович, ми зрозуміли, що там зв'язок у вас втрачається. Ви скажіть мені, висновок позитивний? Скажіть чітко, висновок позитивний, що він є в інтернеті, він оприлюднений, позитивний висновок? Ну, наступного разу прошу більше не покидати університет, коли відбувається така серйозна нарада, тому що ми вас погано чуємо. Можливо, трошечки ваш телефон остине, як колеги мені пояснили, і ви тоді нам ще скажете висновок, скажіть нам чітко українською мовою чи англійською мовою позитивний. Так, все, зрозуміло. Є в нас висновок безпосередньо, все, ми почули, позитивний. Є в нас висновок в спеціалізованій раді, відповідно, він є на сайті, тому ми надаємо слово Джен. Джен, все, пан Бутенко, виключайтеся. Виключайте мікрофон.
I'm not
Okay, just one moment. Okay, Jen, can you give some information or some answers for this review, for Andriy Butenko review, please?
Dear Andrea, thank you for your thorough view and your comments. We agree with them, but let us provide answers and explanations to some of them. My answer of the remark for The author doesn't identify promising breeding material hybrid variety that could be used in further breeding work. The F2 progeny obtained from the crossing between Mexican large spike and Bainon 419 was used to confirm the preliminary QTL molecular marker interval using molecular marker detection, followed by selection of multi-gay phenotypes and functional validation of the candidate genes, and finally, the multi-gay genes obtained were transferred into weight. My answer is finished.
Okay, thanks a lot. Okay. Are you satisfied, пан Бутенко? Ви задоволені відповіді? Головою махніть і скажіть, так, є, чи не є. Так, зрозуміло. Так, наступний наш рецензент Верещагін Ігор Володимирович. So we have other reviews who checked the dissertation, who work in our university. It's Docent Verещагін. Igor, you are welcome. Будь ласка, ви не перший момент, але головний. Review of the official review Verещагін, PhD of Agricultural Science, Associate Professor of the Department of Selection and Seeds, Professor Venturer of SUNY National Agrarian University. Also, PhD ЧЄНЬ Breeding and Genetic Things of Clinical Threats, submitted for scientific degree in the field of knowledge, branch of agriculture, science, specialty 201 agronomy. First one, in events of the dissertation topic, it was the remainder of the last important providing population. Numbers constant growing. Currently, a significant number of breeding programs have been created aimed at the maximizing realization of genetic and adaptive potential of winter wheat. However, at the same time, this is one improvement of the genetic diversity of modern varieties, which significantly limit the possibilities of classical breeding methods in particular selection and hybridization. One of the ways to solve this problem is the use of marker selection, UACAS, and introduction into selection practice of selection based on molecular markers that are linked to target genes. Член work is aimed at solving the problem of identity of the molecular genetic basis of winter wheat for the genetic traits, and particular of the structure of the year, the number of years, and the rate of 1,000 years. The author gives a number of examples of the effective use of molecular markers in selection work. This other plot and concludes that marker selection is promising a significant number of simple sequential markers have been selected, and 143 molecular markers logic have been identified. Yes. In connection of the research, this scientific topic. Research on the topic of dissertation dictates in the following work of the scientific topic of department and selection and feeds after Professor Venturer, developed bioadaptive and ecologically oriented technologies for growing grain crop, taking into account agrotechnical niches and agrobiological concepts of plant growth and development of the condition of the northeastern forest of Ethiopia. Scientific priority and theoretical significance of the dissertation. The genetic mechanism of gray number per spike of it was studied by using the extreme difference of gray number per spike of two parents. Mexico large spike and Taiwan 4199, and was the basis of eliminating the influence of after spike number and 1,000 grain rate of the gray number per spike. Parents and the targeted population, their research results will be more scientific and reliable. Practical significance of the results of the dissertation. Third, author obtained results then can be used in the living practice of clinical. Thus, the genetic profile of the number of grains in the multi-grain variety a lot of these cases to study the main molecular regulatory pathways, genus determination, the number of grains which will contribute to the future study of relevant mechanisms and regulatory methods. The main results obtained personally by the author a map of the genetic linkage of these created, which contains 143 molecular marker lots staining, 19 with chromosomes. These are total genome. Yes, exactly. More detail about your recommendation, about some mistake, about question. Yeah. Okay. The remarks and predictions regarding the term. It is desirable to demonstrate the speed of hybridization of fabric obtained by the speaker. This very economic indicator. When using tables, you need to exactly the name of the table and the user. The name of subdivisions should not be left separately and the end of the table. It's better to start them and in new table. The author does not identify promising bleeding material. Hybrid variety that could be used in the future bleeding material. The small and degradable sequencing research methodologies should be submitted in Chapter 2. The fiber generation must be dedicated to the substrate memory table, not a simple graph. What's about the dissertation? A research director of ЧЄНЬ Breeding and Genetic Tests of Clinical Field submitted for the first two specialized academic council for obtaining the degree of Doctor of Philosophy in the field of knowledge beyond agricultural science and food, in the specialty 2001. The growing according to its relevant scientific and theoretical level, main results of the validity, main provisions and the results published in professional publications and knowledge of the proposal and its practical significance meets the requirement of the Ministry of Education and Cultural Affairs. January 12, on approval of the requirement of the correction of the dissertation at the resolution of the academic committee of Ukraine in January 12, 2022. On approval of the above procedure degree of Doctor of Philosophy and annulment of the decision of the one-time specialty academic council of the institution of higher education, scientific institution, and awarding the degree of Doctor of Philosophy. This ancient institution and cadence with the solution and the cabinet ministers of the time. The content of the dissertation corresponds to the aspect of speciality. I think my I think my review is exquisite. Okay. Nothing for sure is positive, but we saw no review in the website. Okay, dear colleague, please, or not dear colleague. Dear PhD student Jen, can you give some comments for review Igor Vereshchagin? Can you give some answers for these questions?
Okay. Dear Milayevich, thank you for your thorough and professional feedback and valuable comments. We agree them, but let us clarify some of them. My answer about Mark II. The dissertation provides a methodology for construction populations for the study of totals in QTL. However, this methodology is also applicable to other agricultural plants, and in particular to wheat. My answer Mark IV. In April 28, hybridization was carried out using Mexico large spike and binom 4199 to produce F2, F1 generation hybrids in June. Sowing in October 28, marking of each strain in April 29, and collection of F2 seeds in June. That is from an agronomic point of view. They lasted 2 years. The paper also states that data on agronomic traits were obtained for all lines of the 2 generations of Mexico large spike and binom 4199. And then the DNA of all F2 lines was used to screen polymorphic primers. About Mark V, my answer. Why are these the particular patterns from forms chosen? This is because these particular parental forms have unique characteristics: high yields, good resistance to major wheat disease, high gift quality, good adaptability to climatic conditions and soil types, good resistance to drought and high temperatures, and an area length of the growing season. These productive traits were chosen due to their direct impact on yield and overall weight of cultivation efficiency. The number of spikes of the number of gates per spike directly determine the total yield. The greater the number of spikes and
Okay,
gates.
thanks a lot. Okay. Yes, it's because we have not enough time for big discussion. So please stop it. Yes. Okay. So now we must give attention for our review. First exam, it's Mykola Mykolaievich. It was the same question you and Vereshchagin, so it was the same, and Jen maybe give some information. So please read your review. And not biggest one, because I don't know how the temperature now in Poltava, but in Sumy now maybe 35. 35.
Okay.
You see, it's really very hot. So not spend 1 hour, please. Микола
Окей.
Миколайович,
Дякую, Андрію Васильовичу. Я повинен зачитати весь відгук чи тільки discussion clauses and remarks for this dissertation?
yes, you can.
Не почув.
Okay, you can just main point in activity, methodology, and all your questions. And conclusion.
Well, so review of the official opponent for ЧЄНЬ ЦЯОЯНЬ dissertation on the topic breeding and genetic basis of winter wheat ear traits, relevance of the topic and the obtained results. Modern breeding increasingly resorts to another promising method, the creation of almost isogenic lines. This method allows combining a wide range of economically valuable traits and productivity elements in wheat plants, ensuring dynamic yield growth. In this context, specific tasks arise to study the patterns of expressions of major quantitative traits, such as the number and weight of grains and the possibilities of genetic management of these traits. The most significant scientific results obtained by the researcher personally consist in the independent planning and conducting research. The author personally analyzed and summarized the theoretical material relevant to the chosen scientific topic. As a result, a program of experimental research was developed and implemented, which included a cycle of field and laboratory research. The scientific novelty of the obtained results lies in the application of molecular biology and genetic analysis method to achieve specific breeding goals for winter wheat. The genetic mechanism of the formation and inheritance of important traits such as ear productivity and the weight of 1,000 grains were studied. During the dissertation research, they also used quite modern laboratory equipment, which in turn allowed for the investigation of important aspects and the molecular level related to the most economically valuable traits. The practical significance of the obtained results lies in a certain optimization of the breeding process, the mechanism of genetic control of productivity traits, in particular the number of grains in an ear, has been established, which creates for the study of other genetic process. Justification and the reliability of the obtained scientific result. The scientific methodical and scientific technical level of the conducted research is quite high. Application of method of molecular biology and genetic analysis in combination with biometric definitions contributed to the establishment of important regularities in the formation of economic and valuable traits. The role of microRNA in the mechanism of grain formation was established, which allowed the author to make an assumption about the possibility of molecular ways of improving the wheat breeding process. These traits had high variability and were characterized as strong correlation depends. A gene locus has been established that control almost 12% of the phenotypic manifestation of the 1,000 grain mass trait and can increase this indicator. The main results and provisions of the research are published in 2 publications entered into the Scopus Scientific Database, 3 specialist scientific publications of Ukraine, and 7 conference material. In the introduction, there are quite specific, albeit brief, details about the relevance of the chosen research direction, its purpose, scientific novelty, and the specific research method. Chapter 1 consists of several subchapters where the author attempted to adhere to a logical presentation of theoretical material. In clause 1.1, the author describes the localization of quantitative traits related to grain number in the ear and 1,000 grain weight. The chapter includes a relatively detailed description of gene mapping method. Additionally, genes responsible for grain number is in the air, 1000 grain weight, and their interactions with other traits and genes are described in detail. The advantages of using genetic markers in practical breeding are highlighted, significantly reducing the breeding process. Chapter 2 contains a description of the source material, the two varieties that were studied, and brief description of the experimental program. Methods of DNA extraction and analysis of molecular markers are described step by step and is sufficient detail. As follows from the description of the research methodology, they were performed at a high technical level, which made it possible to obtain representative results. Chapter 3 presents the research methodology and literature review related to the study of microRNAs, as well as the research results. The conclusion appear detailed but lack structure. They contain both general interpretation of the role of microRNAs for the development of wheat grain and to clarify their role in molecular mechanism, as well as specific information on the importance and functions of known and new compounds, microRNAs, in the formation of plant organs at the cellular level. Evaluation of the language and style of the dissertation. The dissertation, written in English and heavily laden with scientific terminology, gives the impression of extensive effort. However, its chapters often don't correlate well with the subsequent material presented. There is a desire for a greater number of illustrations which are currently difficult to read. The obtained results are voluminous but lack argumentation, are presented in a not entirely logical sequence and lack interpretation. No mathematical evaluation criteria are provided. Compliance of the dissertation with the specific specialty and requirements. The topic of the dissertation corresponds to the aspect of genetics, molecular and cellular biology, and wheat breeding. Unfortunately, the dissertation lacks sufficient information on the phenotypic expression of quantitative traits, which complicates its assessment from breeding perspective. And discussion clauses and remarks to the dissertation. This thesis gives a contradictory impression. On one hand, the relevance of the topic is indisputable. As a scientific and technical look at the tool it's selected for addressing the research objective allows for discussion. The representativeness of the obtained results and the further prospects of research and scientific implementation. On the other hand, the design, logical coherence and informativeness of presentation, as well as the interpretation of certain points, require detailed clarification. Primarily, this concerns the following points. One page 24, the dissertation connection to scientific topics is described in one word. 2. When discussing genetic difference between parental forms, the author starts the analysis of literature source from tomatoes, which shifts the research focus. It would be advisable to place table 1.1 on page 35. The next field research was conducted during 2 years, but from an agronomic standpoint, they lasted only 1 or 2 years. Therefore, it was necessary to describe in detail the methodology that allowed obtaining representative data, which would also enable formulate representative results. The next, it is unclear from the research methodology why second generation hybrids were chosen. It is mentioned only once that they can provide the fullest spectrum of genotypes, but this is insufficient. Why were the specific parental forms chosen? Why were only the specific productivity traits selected and not others? What are the correlation between productivity elements and morphological traits? Questions arise, such as how all of this relates to yield through correlation. 6. In chapter 3, the presentation of the research methodology begins again, although this should have been done in the previous chapter. The 7th. The presentation of material appears as overlay, concise statements of facts without argumentation and formulation of preliminary conclusions that would allow for the formulation of main conclusions. 8. Figures in the dissertation are presented in a small scale, making it impossible to read most of them, and the reader is forced to rely solely on the author's interpretation in the description. According to the opponent opinions, the conclusions purely correspond to the content of the chapters themselves and hardly corresponds to the proposed objectives of the research program, even the keywords listed in the dissertation abstract, its relevance, and the scientific novelty of the research. Then, point 2 of the conclusions discusses the heritability of quantitative traits, likely grain number in an ear and 1,000 grain weight, but this is not a scientific novelty. The 11th, in point 3 of the conclusions, it is stated that there is a direct correlation of 0.95 between the number of ears or grains and the 1,000 grain weight, but it is unclear why there is a direct and almost functional correlation between traits that genetically have an inverse relationship according to the author's research. 12. A significant number of conclusions, specifically points 4, 5, 7, 9, 10, 18, 19, 22, and many others, simply state individual narrow aspects of the dissertation and don't correspond to the conclusion in assessed. 13. Point 20 of the conclusions doesn't explain what exactly caused the increase in grain weight, a metric that is not even discussed in the dissertation. Is it referring to the 1,000 grain weight? Next, the recommendation based on the research result don't correspond in essence to the concept of recommendations themselves, don't arise from the content, purpose, and objectives of the dissertation research. They merely describe the research purpose and possibilities once again. The motivated comments and questions require detailed explanation and answer, as well as substantiation of points during the public defense. General conclusion. Dissertation work by ČЄNь ЦЯОЯНь titled Breeding and Genetic Basis of Winter Wheat Traits, submitted for public defense of obtaining the degree of Doctor of Philosophy in the field of knowledge, agriculture, science, and food in the specialty agronomy by its relevant scientific and methodological level, obtained and the published results meet the requirements of the order of the Ministry of Education and Science of Ukraine, number 40, dated January 12th, on approval of the requirements for the dissertation formatting. And the resolution of the Cabinet Minister, ну, далі не буду читати, вже прочитати. On approval the procedure of awarding the degree of Doctor of Philosophy and cancellation of the decision of the one-time specialized academic council of a higher education institution, scientific institution, on awarding the degree of Doctor of Philosophy as amended by the resolution of the Cabinet Minister number 341. The author of the dissertation, ЧЄНЬ ЦЯОЯНЬ, desires to be awarded the degree of Doctor of Philosophy in specialty agronomy. Thank you.
Okay, thank you very much. Mikola Mykolaievich, you every time make very deep review. Okay, it's very good. So, ЧЄНЬ ЦЯОЯНЬ, please give some comments or some answers for review Mykola Marenovich.
Dear Mykola Mykolayevich,
Yes.
thank you for your thorough and professional feedback and valuable comments. We agree with them, but let us clarify some of them. Question one, my answer of the question one. The dissertation provides a methodology for constructing populations for the study of tomatoes in Qatar. However, this methodology is also applicable to other agricultural plants and in particular to wheat. My answer of the question two. In April 28, hybridization was carried out using Mexican large spec and Binom 4199 to produce for F1 generation hybrids in June, sowing in October 28, marking of each strain is April 29, and the collection of L2 seeds in June. That is from an agronomic point of view. This lasted 2 years. The paper also stated that data on agronomic traits were obtained for all lines of 2 generation of Mexican large spec and Binom 4199. Then the DNA of F2 lines was used to screen polymorphic primers. My answer of the question 3. This is because this particular parent forms have unique characteristics, high yields, good resistance to major wheat disease, high grape quality, and good adapted to climatic conditions and soil types, good resistance to drought and high temperatures, and on every length of the growing season. 4. My answer of the question 4. The greater the number of specks and gaze, the higher the yield per square meter of crops, which is the perennial goal of breeding new varieties. These trees also allow for a quick assessment of the economic efficiency of the variety. Varieties with a large number of specks and gaze usually have better resilience to adverse conditions, such as drought or poor soil conditions, ensuring stable yields in various climatic zones. These characteristics are easy to measure and assess in the early stages of breeding premise. 5. My answer of the question 5. The correlation between productivity elements and morphological traits in winter wheat are crucial for understanding and improving breeding programs. Results of the correlation analysis show that the number of gaze prospects positively correlated with a greater number of prospects had a significant level. The number of the gaze prospects had a positive correlation with plant height and spec length. The weight of 1,000 gaze had a positive correlation with the gaze size and a negative correlation with the number of gaze prospects. Spec length has a positive correlation with the number of gaze prospects. And the weight of 1,000 gaze the number of productive tillers had a positive correlation with the number of spikes per plant and the weight of the southern gates. My answer of the question 6. Correlations provide important information about how these traits affect yield. For example, positive correlation will indicate that if the number of gates per spikes increase, this is often associated with a greater number of spikes per plant, directly increasing the novel overall mass of the yield. Taller plants can absorb more gates, and longer spikes can accommodate more gates and may contribute to increasing the average gate weight. Large gates increase the weight of some gates, and a higher number of the productive mean more spikes, all of which contribute to increased yield. My answer of the question 7. We wanted to explain in more detail that the scientific novelty of this study is to use our molecular biology tools to study the heritability of quantitative traits, such as the number of the gates per year and the weight of certain gates. In particular, polymorphic primers were used for the first time to screen the DNA F2 lines of the Mexican large year and binom 4199, which allowed us to identify genetic markers associated with these traits. My answer of the question 8. It is noted that there is a direct correlation of 0.9563 between the number of spikes and the weight of some gates, which means that these traits are strongly related to each other. Secondly, the research methodology, including the accuracy of measurements and statistical methods used, could also have influenced the results. Thirdly, we can't rule out the possibility of complex genetic mechanisms such as plantropy. When one gene affects several traits, such can lead to a high correlation between them. It is also important to keep in mind and that data obtained may be part of a more general trend that manifests itself in these particular research conditions. My answer of the question 9. And in the conclusion to the paragraph 20, it is not entirely clear what exactly caused the increase in gate weight. It should be clarified that in this case, we are talking about the weight of certain gates. However, we haven't provided sufficient detail to explain the reasons for this increase. For more complete understanding of the reasons for the increase in weight per certain gates, additional factors such as genetic characteristics of varieties, growing conditions, and possible interactions between genetic and agronomic factors should be taken into account. In our future research, we will definitely look at these aspects in detail to get a clear picture of the mechanisms that influence this indicator. My answer is finished.
Okay. Mikola Nikolaevich, are you satisfied? It's a really big discussion, many questions, many answers.
I think I can to be satisfied. Thanks. I'm satisfied.
Okay. Thank you very much. Okay, we have one more additional review. Leonel Olovich, you are welcome. Please read main point of your review, the main questions for PhD students.
Dear colleagues, I agree with your high opinion about ČEN's work. So let me focus only on discussion point and acknowledge. The abstract mentions the combination Mexico very large colossus with binom 419, the second chapter Mexican large spike with binom 419, and the third chapter refers the variety binom 4199. If Mexico very large colossus and Mexican large spike and binomial 419 and binomial 4199 are different samples, then the structural elements for the thesis are not related to each other. If we work only with one pair of parents, we must have all information about it. The abstract in Ukrainian is sloppy, some sentences are unclear. From table 2.2, it is unclear what the values of mass of 1,000 grains and the numbers of grains per year of the parental forms, and whether they differ significantly or not significantly. And as I think there are many technical mistakes. For example, the paper discussion QTL related to the number of grains per year, but page 83, 84 are about QTL related to the number of year. The weight of the kernel weights denote either TGW or TKW. It is better to use more commonly used F2 in subscript instead of F2, and MUL in Greek instead of UL. There are necessary space in some place and no space where they are needed. Some sentences are completely repeated or some sentences incomplete, and so on and so on. The poor resolution of many of the figures does not allow to extract information from them. The above comments and suggestions cannot adversely affect the conceptual provisions of the dissertations and the scientific value of the original research work. General conclusion: the dissertation work of ЧЄНЬ ЦЯОЯНЬ, molecular genetic basis of winter wheat traits, submitted for defense in terms of its relevance, scientific and theoretical level, made result and validity, main provisions and the results published in professional journal, knowledge of the statement and practical significance meets the requirement of the order of the Ministry of Education and Science of Ukraine, and its author, ЧЄНЬ ЦЯОЯНЬ, deserved to be awarded degree of Doctor of Philosophy in specialty 201 agronomy, field of knowledge 20 agricultural science and food. Thank for your attention. А де всі подівались?
Okay, just one moment, because our secretary will bring some water, because very, very hot now, maybe 40. So we can finish. Jen, you have possibility to give answer for questions of Oleg Leonov, please.
Dear Oleg Yulievich, thank you for your remarks. We agree with them, but let us respond and explain some of them. Amongst of the question 1, we apologize for the misunderstanding. There was a technical error. These are Mexican large spike and binomial 4199 samples that we use in our research. Amongst of the question 2, let me clarify. In table 2.2, the mean values of the number of gays per year, and southern give it of Mexican largest spike are 100.03, are 103.3, and 42.85 respectively, while the mean values of the number of gays per year and southern give it of the Bainon 4199 are 51.3 and 49.15 respectively. That is, they differ significantly in terms of the number of gays per year and southern give it, which is indicated in the table by one symbol for difference and two symbol star for very significant difference. My answer is finished.
Thank you very much. Are you satisfied? The
Yes, I
answers?
am.
Okay, thanks a lot. Okay, dear colleagues, now we must start discussion. Please, okay. We must start discussion. We can discussion in English or sometime in Ukrainian. So about PhD, about methodology, about rules, mistake in dissertation, about presentation. So everything, everybody must take part in our discussion. So maybe Nikolaevich, you have good experience in this meeting. Please take part in discussion. Not too much, maybe 5 sentences possible.
Andrei Vasilyevich, dear colleagues, the dissertation made a contradictory impression, and my review is the same. I must say that at first I didn't like this dissertation. What's the main issue here? First, the breeder works with a phenotypic value of indicators, but here we don't see it. We are dealing with molecular genetics. But that isn't the main thing. The point it is that the beginning and methodology of the work aren't related to the conclusions. The literature review and experimental parts had its own life and its own sequence. I would say that it isn't known where the proposition for further use in selection come from, and are they proposition at all? Where do the conclusions come from? Each part of the works has its own rationale. I had difficulty with my own conclusion. I thought it was some sort of competency test. Will the opponent give a positive conclusion or a negative one? On the one hand, the dissertation has a high methodological level, was performed at a high instrumental level, and interesting data were obtained. This is my subjective opinion. On the other hand, formal requirements may have a negative impact of subsequent studies. The author may refuse further research, and the achieved results may be lost, but these results may have a scientific perspective. I hope that in the future she will present her own research more logically. That's why I decided to give a positive review. Thank you.
Okay, thank you very much. It's really very deep conclusion and review. Okay. Oleg Yuryevich, please, your opinion about this dissertation, about this PhD student.
Yes, thanks. The dissertation topic, breeding and genetic basis of winter wheat ear traits, addresses important aspects of the breeding and genetics of the essential grain crop. There are many trials for studying of winter wheat, such as disease resistance, lodging resistance, frost resistance, many parameters of grain quality, but yield and productivity are the main triad for winter wheat. ЧЄНЬ has analyzed the genetic mechanism influencing the development and productivity of the winter wheat spike, proposing new approaches to increase its yield. The candidate's professional characteristic influence extensive knowledge in plant genetic, skills in modern breeding and biotechnology methods, as well as experience in conducting comprehensive scientific research and data analysis. Therefore, I believe that ЧЄНЬ ЦЯОЯНЬ deserved to be awarded the degree of Doctor of Philosophy in the specialty 201 agronomy. Thanks.
Okay, thanks a lot. Let's read out a deep and deep review. As about me, so I agree with Mikola Mykolaievich Marevich and with Oleg Yulievich Leonov, because it's really the biggest scientific in Ukraine with winter wheat. So they know more deeper in our commission. So I know why they're worried about some mistake. So we must discuss, it's very clear, because it's really very interesting and very deep job. Maybe something not the same like in Ukraine, because it's really China. But one of them, what I think is really good scientific work, because they use a very high level equipment which can give really clean conclusion. It's not positive point. Next positive point, it's about article. So we have 2 articles in very high level journal. It's really also plus. So I think, yeah, it's really very hard to tell everything clear, because not all PhD works, it can be everything clear 100%. It must be, it can be, have some mistake. In these works, I agree with reviews again, have many mistakes, like grammar mistake, like, something happened in a small mistake in methodology. So, but I'm sure in future, this young scientific will make new conclusion and maybe more clear present and more clear printed your dissertation. So I think it's a quite good level investigation with small mistake which need change in the future. So my review, I think the ЧЄНь ЦЯОЯНь can be have the PhD in 201 agronomy. So it's my opinion. It can be, I think everything must be good. Okay, what about Andrei Alexandrovich Butenko? What's your opinion?
Thank you. Dear colleagues, the dissertation by ЧЄНь ЦЯОЯНь, Breeding and Genetic Basis of Winter Wheat Earth Traits, illuminated key aspects related to the development and refinement of modern selection method aimed at enhancing the yield of this valuable crop. A mapping population and genetic linkage map for wheat were established. This finding not only our
Андрій Олександрович, мікрофон включіть.
Включив, то паралельний дзвінок. This finding not only deepen our understanding of the molecular mechanisms underlying productivity traits, but also contribute to the development of more effective selection methods for consistently increasing winter wheat yields. The dissertation's material has been fairly articulated in 12 scientific publications, including 2 articles in Scopus Indexes journals and 3 articles in Ukrainian scientific publications. These dissertations represents a significant contribution to the advancement of agronomic science and holds practical implications for the modern agricultural sector. Based on its relevant scientific theoretical level, substantive results and novelty, as well as the practical significance outlined in the specialized publications. It meets the requirements set forth by the Ministry of Education and Science of Ukraine regarding dissertation formatting and the procedure for awarding the degree of Doctor of Philosophy. Therefore, I recommend that ЧЄНЬ ЦЯОЯНЬ be awarded the degree of Doctor of Philosophy of the field of 20 agricultural science and food production, specializing in 201 agronomy. Thank you for your attention.
Thank you very much, Андрій Бутенко. Very good conclusion. Okay, Верещагин Игорь, you are welcome. Thank you. I remember when ЧЄНЬ ЦЯОЯНЬ Николаевна in our laboratory of PCR diagnostic with our equipment like German cycle, UV box, electrophorex equipment, and I know for knowledge and skills of our student, ЧЄНЬ ЦЯОЯНь. The work of research was very, very deep by molecular biology technologies, using molecular markers filter between selectivity. This is a very perspective technology and very useful. But this is I think this is a strong side of this research. And what about my conclusion of this work? I think the dissertation work of ЧЄНь ЦЯОЯНь building on genetic causes of winter wheat traits contributed to a difference in the specialized academic councils for obtaining the degree of Doctor of Philosophy in the field of knowledge, 20 agricultural science, and in the specialty It's 201 agronomy according to the relevant scientific and theoretical reason. Main results of validity, non-validity, and the results published in professional publication. Okay, thank you very much, Igor Volodymyrovich. And now in our discussion can take part the scientific tutor Natalia Mykolaievna Tandiba. Please note for 1 hour. Please very short about PhD student, about Xiaoyan chain. Yes, please switch on your microphone. Включить микрофон. Natalia Mykolaievna, switch on your microphone. Yes.
Thank you
And
so much.
please
Зараз
make
мене чутно говорить?
short. Yes.
Okay, I try. Thank you. Dear Chairman, dear members of the Dissertation Council, dear participants, let me now read out my entire response, but rather focus on the main points. About general characteristic of the applicant, I can tell you that during research, ČÉNЬ proved to be a thoughtful, thorough experimenter. Despite the large amount of planned, she was able to successfully complete the research tasks due to her hard work and diligence. Throughout her postgraduate studies and beyond, she has mastered all general and specific research methods and proved to be punctual, persistent, and responsible person. The educational and scientific program was completed by the applicant in full. A large amount of field and laboratory research was conducted. The same large amount about scientific literature of the topic was analyzed, which was ensured by a significant amount of the experienced material, correctly selected research methods, and considered analysis of the experimental data obtained. I believe that ČÉNЬ will be worthy of the title of scientist. I would like to wish you, ČÉNь, confidence and success in your endeavors. Get your PhD sooner and find you the job you would like. And thank you so much, dear colleagues, for your attention and your huge support. Thank you.
Okay, thank you very much, Наталя Миколаївна. Шановні колеги, ви зараз... If somebody won't take part in discussion again, maybe some people from PhD office, maybe some people from ČÉNь. Okay, nobody, yeah? Everybody who wants make it. Шановні колеги, всі, хто хотів, прийняли участь в дискусії. Дискусія дійсно глибока, питання є, на то воно й наука. Маємо рухатися в міжнародному просторі, тому маємо такі виклики. Зараз переходимо до обговорення проекту рішення нашої спеціалізованої вченої ради і дозвольте його зачитати державною мовою. Отже, спеціалізована вчена рада Чемського національного аграрного університету, Міністерства освіти і науки прийняла рішення про присудження ступеня доктора філософії з галузі знань 20, аграрні науки та продовольство на підставі прилюдного захисту дисертації селекційно-генетичні основи ознак колосу пшениці озимої за спеціальністю 201 агрономії. ЧЄНь 1989 року народження, громадянинка Китайської Народної Республіки, освіта вища, у 2018 році закінчила Фінансовий інститут наук і технологій Китайської Народної Республіки, яка здобула освітній ступінь магістра за спеціальністю генетика і селекція. Аспірантуру закінчила в 2022 році Сумському національному аграрному університеті Міністерства освіти і науки України, місто Суми. Роботу виконано в Сумському національному аграрному університеті та Фінансовому інституті науки і технологій. Науковий керівник Кандиба Наталія Миколаївна, кандидат історичних наук, доцент кафедри селекції насінництва імені Миколи Дем'яновича Гончарова Сумського національного аграрного університету. Здобувач має 12 наукових робіт за темою дисертації, з них 5 статей, 3 – це фахове видання країни і 2 занесені до міжнародних баз даних ОКУС і 7 тест доповіді міжнародних наукових конференцій. Таким чином, ми маємо всі підстави перейти до процедури відкритого голосування. Прошу всіх членів спеціалізованої вченої ради визначитися, проголосувати і озвучити своє рішення щодо присудження ЧЄНЬ ЦЯОЯНЬ титулу доктора філософії за спеціальності 201 агрономія. Тобто, називаю прізвище, ви кажете голосно за чи проти. Отже, Мельник Андрій Васильович, за. Бутенко Андрій Олександрович,
За.
Верещагін Ігор Володимирович, за. Маренич Микола Миколайович,
За.
за. Леонов Олег Юрійович.
За.
Дякую, шановні колеги. Отже, за. Ми фіксуємо в нашому протоколі 5, проти 0. Таким чином, на підставі результатів відкритого голосування спеціалізована вчена рада присуджує ЧЄНЬ ЦЯОЯНЬ ступінь доктора філософії в галузі знань 20 аграрні науки та продовольства за спеціальністю 201 агрономії. Шановні колеги, дозвольте привітати здобувача та її наукового керівника з захистом, побажати проходження успішного підтвердження Міністерства освіти документів, а нам усім відповідно велика подяка, що ми в такий важкий час провели дане засідання. І кінцеве слово надається здобувачі. Вона може сказати декілька слів. So, ЧЄНЬ ЦЯОЯНЬ, we would like to congratulate you and your tutor with successful defense. So, everybody, so 5 члени комісії, member of our commission shall agree. So, you have time to give some word, some information. So, your time for your small speech from you.
Dear Chairman, dear members of Specialized Council, dear audience, allow me to express my sincere gratitude for your attention and support during the defense of my dissertation. Today has become an important milestone in my life. I'm grateful to each and everyone for you and for your cooperation, advice, and constructive criticism that helped me achieve this goal. I would also like to express my sincere and gratitude to the university administration for the opportunity to study, and to the university's graduate school for their constant support, to the entire staff of the Professor Gartenloff Department of Plant Breeding and Seed Production, and especially to my supervisor, Natalia Kanadiba, for her patience, constant support, and help. My journey into the world of science has been interesting and filled with the study of various aspects of agronomy, and therefore, I have a great desire to continue my scientific research. Thank you again for your time, attention, support, and cooperation. Thank you.
Okay, thank you very much. Да, така важкий захист, це згідно чинного протоколу ми витримали. Позитивний є результат. Ще раз дякую, шановні колеги, за те, що ми конструктивно провели дану спеціалізовану вчену раду, вказали на ті моменти, які треба покращити, і прийняли ці колегіальні рішення. Всім дякую, всім нам мирного, переможного неба. Ну і слава Україні!
Героям
Слава!
Так, дякую. Все.